We welcome Julia Erk. She's a PhD student at the lab of Joachim Wegener at the University of Regensburg. And today she's going to present about her experience with monitoring GPCR signaling and combining it with photochromic ligands using label-free impedance assays.
So actually, I'm more on a fundamental research part that I'm working on. So I'm doing my PhD at the moment. So it will be a little bit more basic research. So not so much application oriented, but in general, signaling cascades are all around us. So it is always important to understand what is really going on inside a cell. So whenever we have a morning coffee or we feel the feeling of hunger, then there are always some signaling pathways involved. And also signaling is it's really crucial to understand what is going on, because a lot of drugs and in pharmacology, it is really necessary to really understand what's going on. And we wanted to combine the GPCR research with the device that we had from Nanion. So on the one hand side, we use the cardioexcite, and on the other hand, we use the Atlas system. And I would just like to show you today how we use it in our lab with a combination of photochromic GPCR ligands and those two systems. So we'll just walk you through the protocol that we're using and what we are doing and what we are investigating. So I would like to get everyone on the same page here. So I would just give a short introduction about G-protein coupled receptors, since I learned that a lot of you are mainly working on ion channels. So I would just like to explain what G-protein coupled receptors are. So they are basically one of the most important receptor classes that there are. They are the target for about over 30% of FDA-approved drugs. So they have really high clinical relevance. And also, there are different pathways, as you also saw in the previous talk. So we have the G-alpha I pathway, the G-alpha S pathway, the G-alpha Q pathway. And what basically happens is that we have a receptor that is located in the cellular membrane. It has those 7 transmembrane domains. And whenever a ligand binds on the action cellular site, then the G-protein coupled receptor becomes activated. The G-protein dissociates from the receptor, and then it targets downstream signaling effector proteins. And in my case, I use the NPY Y4 receptor as a model system, just to show you some data on this one. This is relevant for cardiac diseases, and it's also linked to appetite and obesity. And this one is basically G-alpha I coupled. So this means whenever the G-protein coupled receptor is activated, it acts on the adenylate cyclase. The adenylate cyclase is an enzyme that forms cyclic AMP, so one of the most important second messengers inside the cell. And it acts in a way that it inhibits the adenylate cyclase. So whenever the receptor is activated, then the cAMP levels inside the cell decrease. And then after this whole signaling happens, the cell changes its morphology. And this is what we can then read out in the end with the impedance. And since yesterday I talked to some people and they were not quite too familiar with the impedance technique, but more with the patch clamp technique, I would just like to give you a short overview of what we are measuring actually. So we are seeding the cells on a substrate that has 2 different gold film electrodes on the bottom. So as you can see here, this is one plate for the cardioexcite. So we have those 2 electrodes, one larger counter electrode and a small working electrode, and then we seed our cells on top of them. We apply a small non-invasive current. This is really important, so it's a non-invasive technique. And then the current is forced to flow either around the cells or couple through the cells, depending on the frequency that is applied in the end. And then whenever we do the GPCR stimulation, the cells change their morphology, and then we can detect it in an impedance change. So, yeah. Okay. When we then do the stimulation, we can do it with a simple ligand, so an endogenous one. For the NPY receptor, this is just, for example, the HPP, which is the human pancreatic polypeptide. And if we do it in a concentration-dependent manner, then you see that whenever we stimulate the cells at time point 0, the impedance goes up. So this means that we can correlate that whenever the cAMP inside the cell is lower, for these CHO cells carrying this NPY Y4 receptor, the impedance goes up. So low cAMP levels, high impedance. And this can also be confirmed when we go one step further down the signaling cascade. So when we apply a cAMP derivative directly So in this case, we use the 8-CPT CAMP. It is basically a membrane permeable version of the endogenous CAMP. And we see the response turn the other way around. So whenever we add the derivative, then the impedance goes down. So higher CAMP levels, lower impedance. This is all good and nice, and it works pretty well and pretty reproducible. But the problem is that we still have some liquid handling. So whenever we add it at time point 0, we disrupt the cell layer, and we get those bumps, as you can see here. And also studying those GPCR responses is then diffusion limited because of the liquid handling. We add the ligand on top, and we have to wait until it diffuses to the receptor. So what we thought about doing is to combine it with photoswitchable bioactivity. As you already heard in Mr. DeWard's talk earlier this morning, it is basically that you have a molecule that comes in 2 different isoforms. So for our case, we have a GPCR ligand. This is just an example. So we have a peptidic ligand. And the special thing is that here you have this photoswitchable moiety installed. It's also an azobenzene. Those work quite well. They are pretty stable and pretty easily switchable. And then you apply it to the cells, and you can also have it the other way around. So here you see the cis version, and here you see the trans version. And then you apply it on top of the cells. And the special thing is you irradiate with a light of a specific wavelength. And then you can toggle between those 2 isoforms. And this now gives us the advantage that we just add a ligand at one time point. We leave it on top of the cells. We irradiate the cells with a ligand on top. And then we want to activate the receptor only with light, so no liquid handling anymore. And then we want to read this out with the impedance. So the regular way to do those GPCR assays is that you can do optical readouts, like you have a mini-G protein coupling, or you have radiometric readouts, but they have the drawback, of course, that radiometric is super toxic. And with the optical readouts, you often have the problem that you switch your photochromic ligands on accident because they are based on an optical readout. And then you have to think about the interference between the readout and the actual switching of the ligands. You can also do this electrochemically or even impedance-based. And for the other assays, as I mentioned earlier, it is often the case that they target only one single pathway. So only, for example, if you do the mini Q recoupment, then you only have insight on one part of the pathway and a non-holistic readout. But for the impedance, you get like an overview of everything that is going on inside the cell. And also most of those conventional assays are endpoint assays, but we want to monitor the activation of the GPCR in a reversible manner. So this means we have to do it online and we have to measure continuously. And this is also what is then possible with impedance. So here you can see basically the setup that I'm always working with. So it is not so much high throughput, but I have to do it all on my own. So this is what the measurement setup looks like. I work in the dark because, of course, I cannot allow the ligands to switch on accident. So I have to work with this wonderful red LED, as you can see here. And then I work with a cardiac site or maybe even the Atlas system. And the procedure is basically always the same. So I seed the cells on day 1. I exchange the medium on day 2. And this is just to ensure that I have reproducible expression of the receptor and that the cells are healthy. And then on day 3, I do the actual measurement. So I switch to a measurement medium. Then I add a pre-stimulation. So in this case, it's forskolin. Maybe you already read it in the talk before. So forskolin basically stimulates the adenylate cyclase. So this means CAMP goes up. Then I stimulate the GPCRs, CAMP goes down again. So this just gives me a higher signal change when I do the stimulation of the GPCRs. And then I do the actual measurements. So dose response, irradiation, overshown experiments. And now let me show you some data. So what I always do first is I record dose response curves for both isomers. So in this case, on the left-hand side, you can see the trans isomer, and on the right-hand side, you see the cis isomer. And if you now compare the same concentrations, for example, with the red or the orange curve, as you can see here and there, you can already see by naked eye that there is a difference in the activation potency of those different ligands. If we then do area under the curve calculation, we can get some nice dose response curves. So we can plot them in the same graph, and you see that the cis isomer is more potent than the trans isomer. And then the next step is to search for a concentration where the difference is most pronounced between the two isomers. Yeah, and this is then this range here. And from this on, we can do then the actual switching experiment. So this is basically our end goal then, to have a system that allows the detection of the GPCR activation in a reversible manner online. So what you see on the left-hand side is that in blue, you see the cis isomer that was kept in the dark. So I basically put aluminum foil on top of the wells that I did not want to be irradiated, and I irradiated all the other wells. And in purple, you see the trans conformation also kept in that dark, and the cis conformation that was then switched, where you can see the light bulbs, and you see at those gray lines that you can switch it with one wavelength towards the one isomer, and then you switch it with the other wavelength towards the other isomer. So it works in both directions, and it works reversibly. And this is then also true the other way around. So when you start from the trans conformation, and you go towards the cis, and you go towards the trans isomer. And so we showed that in the end, we can really activate the G protein coupled receptor only with the help of light, without any liquid handling, allowing us to further study what is really going on during the signaling cascades. And of course, in the far future, it would be a goal to have then medicine or molecules that can be switched on and off inside the body with simply the help of visible light. And with this, I'm actually already at the end. I would like to thank my supervisor for always being there for me and being really kind and giving me this nice topic. And then also I would like to thank the person that synthesized those ligands, actually, all members of my working group, Lanyon for the nice technology, and you for your attention.
Thank you very much, Julia.
Very nice talk, Julia. Thank you. What cell types did you use, and have you tried to, is it overexpressing GPCR, and have you tried like endogenous or other cells?
Actually, I just tested CHO cells, and they always overexpressed the receptors, but I tested different receptors. For example, we also did some on the dopamine D2L receptor that also worked quite nice, but I mainly work with overexpressing cells and only CHO cells. So this should just be like a model system that we see that it works.
Maybe one also like technical question. So the so-called L15 medium that you use for the cells, is this any different from a normal like maintenance medium of CHO cells?
Yeah, so it does not have all the nutrients and all the necessary stuff. So it's, for example, it also says no serum. And this is important because the serum, it contains some proteases that would then in the end destroy our ligands. And also it is colorless, so it's also good for our application. But they live well for like 1 or 2 days, but then you would have to supplement